Good afternoon, everybody. I welcome you back for the part 2 of the ALGAE. And before just moving above, so I just want to revise what we did in the part 1. In part 1, we did the types of the algae. There we did the aquatic algae, parasitic algae, and banthophytes, fluviatile algae. Then we have epiphyllophytes. So then today we are doing the other in league of that is endophytic algae, which live within or among the cells of other plants. The example is nostoc, which we did 2 days back in the thallus of bryophytes, that is anthoceros, right? So next is the Anabina cycadacerum, that is the corollary root of cycas, and then Anabina azoli in the azolla water fern, and one more, that is Coleochaete nitellarum. So it is present in the walls of algae nitella, right? So that's why a species name. So then I already did this, the presence of the paranites. Paranites is included by the starch sheath in most families, right? So these starch sheath, when incomplete, is called polar paranite, right? So then I told you there are 3 mesh cells. This is largest, this is smaller than this, and this is the smallest one. So now I am coming to the today family, that is the Xanthophyceae. Xanthophyceae, A means it has a chlorophyll A, and then beta carotene, then lutein. The storage product is chrysolaminarin. So chrysolaminarin is a storage product which I again revise when I do the next family. So chrysolaminarin will remains with us. Vaucheria is mostly freshwater habitat, and the thallus is branched, right? And sonocytic. It's very important. And it is unseptate, means multinucleate, tubular filament. At the base of thallus, a branched rhizodal system, hapteron, right, which attach thallus to the substratum, and the chloroplast is discoid, and which is divided of paranoid bodies, which I showed you here in case of Chlamydomonas, right? So in case of Vaucheria, when I did the introduction, I just told you there are the genera where paranoid body is absent. Vaucheria is their one name. And then we have the Vaucheria amphibia. It is an basically an amphibious species, and then Vaucheria johnsoni, this is on the snow. Last time we also did the one on snow is the Chlamydomonas nevis. So it has hematochrome and it gives red color to the snow, right? So this is the what we have done. So because of this, they are aseptate, but septa are formed at the time of reproductive structure formation. This is very important, right? So they are aseptate, but septa will form when we start formation of reproductive structure. So now come to the reproduction. So reproduction we will have, first is vegetative, is fragmentation, in which the different fragments will form, and these fragments they can give rise to complete plant. Then asexual reproduction is by means of a zoospores. So zoospores here are multiflagellate and multinucleate. It differs from the other algae, right? So these two characters, it differs from the others, and zoospores are known also as syn zoospores because it is multinucleate, right? Or they are also called soinozoospores, right? And second by aplanospores. Aplanospores are also multinucleate, and they are club shaped, and they are non-motile, right? These questions will come, right? They develop in dry condition in terrestrial forms. And the third is hypnospores or cyst. Hypnospores are thick-walled multinucleate segments, right, which divide, producing thin-walled cyst, right? Cyst germinate, and its protoplast come out in form of amoeba, that is amoeboid fashions, and becomes rounded and develops into segmented thallus, which gives like a look like a Gongrosira. I told you when we did that time, one alga will resemble Gongrosira, that is Vaucheria. Hence, it is this stage is called Gongrosira stage. So I'm coming back to the what we did is first is the thallus. The thallus is sonocytic, right? Multinucleate. No septa is there, but septa formation I told you only at the time of this reproductive structure like this. See this septa, right? These are, I told you, basically syn zoospores, which are multinucleate, right? And here the septa will form, right? Here it's very interesting feature occurs. First the vacuole, then the all nuclei move towards the tip, then septa will form, then reverse will occur, right? So reverse will occur in which there is a gradually basically movement of nuclei and chromatophores in the reverse direction, right? See, these are the aplanospores. Aquatic species which commonly produce these zoospores can be induced to produce aplanospore by keeping them in dim light, right? So those aquatic species which produce zoospores, but they have been forced to produce these aplanospores in the dim light, right? So here I told you then there is a formation of these amoeba like called Gongrosira stage, right? So next we are coming to the sexual reproduction part C. So before moving it, I will explain you the sexual reproduction part. So sexual reproduction part is basically oogamous, and most of the species are homothelic, means freshwater, and some are heterotelic, so which are dioecious. It is only in case of dichotoma. It's very important species, right? It is protandrous. In during bryophytes, we also use this term protandrous. The antheridium develops first and oogoniums afterwards. The antheridium, as I showed, just to show in the diagram, they are produced on the main filament or its lateral branches, a curved cylindrical structure, right? Curved cylindrical like this. Curved, right? Curved cylindrical structure. A large number of chromatophores and multinucleate, that is nuclei, accumulate in curved part. So here all they accumulate, and which cuts off by the means of septum, which I already told you. Septa, it is aseptate, but septa is formed only at the time of the formation of reproductive structures. The nuclei of antheridium divide mitotically. So just remember, it is a bit different, mitotically, and form biflagellate enterozoids. Each enterozoid is spindle-like, and insertion of flagella is lateral. When we will see, it is here, lateral, like this. So we are coming to the oogonium. In homothelic species, a colorless mass of cytoplasm as a small protuberance at the base of antheridium. As I did here, at the base of antheridium, a small protuberance like this. And this small protoplasm is called Vanderplasm. So this small protuberance at the base is called Vanderplasm. It gradually increases in size and form a ovoid oogonium, right? So as it is oogamous, oogonium and antheridium, which give rise to enterozoid, they fuse to form oospore. So it is very important to note here that zygote is green in color initially, but it gradually results into the red, right? and due to degeneration of chlorophyll, right? It differs from chlorophyce in chlorophyll absence, B absence of pyranite, I told you, and oil as a reserve material, whereas there we have the starch, right? So then we are coming to back again to this structure. I told you this is the curved structure, and here we have the protuberance called ventriplasm, and it moves up and move down, and then there is a fusion of the cells, right? And it results in the formation of zygote, which I already told you. So now I'm just moving on to next part, that is the ectocarpus, which belongs to the family Phaeophyceae, right? So in case of ectocarpus, there is fasciculatus, which is epizoic, as it is present on the fins of fish. And ectocarpus arabiscus is epiphyte on the leaves of sargassum. Then there is one Macrocystis pyrifera, which is 60 meter tall. It is called giant kelp. The pigmentation, as we read ectocarpus, A is there and then C is there, and C shows we have the pigmentation of chlorophyll C. Then with that, besides this, that is beta carotene, fucoxanthin and viola xanthin, and the reserve food is laminarin and D-mannitol, right? And the plant body is heterotrichous, which is prostrate and erect, and the chromatophores here are plate-like chromatophores with pyranite, and the brown color as Phaeophyceae called brown algae, which I just told you. So it is due to the presence of a pigment called fucoxanthin. See, the refractive bodies, there are other question comes, the refractive bodies present in cytoplasm called fucoxan vesicles. These fucoxan vesicles are the refractive bodies which are present that contain fucoxan, right? Reproduction is first is the asexual reproduction by zoospores, and in zoospores we have first is the unilocular sporangia. These sporangia develop singly at the tip of small branches, and mind it, the first division of nucleus is meiotic, and followed by mitotic divisions, right? So then it has 64 nuclei will form, and they will form zoospores. As it is meiotic, it is uninucleate and pear-shaped and biflagellate, and because of that division, it is a haploid gametophytic plant. Whereas plurilocular, which are elongated, they produce from the terminal cells of diploid thalli. The nucleus divides mitotically. Because they divides mitotically, the zoospores are formed are diploid and biflagellate, and it gives rise to diploid sporophytic plant. Both are physiological. Basically, these development of both unilocular and plurilocular sporangia is affected by temperature and salinity, right? In case of siliculosus, it produces unilocular sporangia at 13 degree, plurilocular at 19, and if temperature is at 16, it produces both unilocular and plurilocular, right? So now we are coming to the next is the tetraspores. So this question also raises, tetraspores are present where? I already taught you the tetraspores earlier also. So diploid plants, they give rise to give rise to 4 tetraspores, and these 4 tetraspores germinate to give 2 male and 2 female plants, and it is present in the dictyota. Now we are coming to the sexual reproduction. Sexual reproduction is oogamous in basically ectocarpus paddy, whereas in others it is gametangia and gametes. In these, I isogamy species, the fusing gametes are similar morphologically and physiologically. And these two fusing gametes are called isogametes, and they fuse to form a zygospore, right? But in physiological anisogamous species, the female gamete settles down about 5 minutes after liberation and secretes a volatile sexual hormone called serotonin. Right? Very important. And the question comes a lot of time. During fusion, several male gametes cluster around it. Like if this is female, and a lot of the male gametes, they cluster around it, and they will form a clump formation, right? In ectocarpus, both gametophytic and sporophytic generations are morphologically similar. That is why such life cycle is called isomorphic. Very important. This question will come. Right? So, this way finishes the brown guinea algae. And now we are coming to the third and the last family, that is Basilerophyceae, a very important family, and is also known as the diatom family. It is Diatomophyceae, and it forms are known as diatoms and called jewels of plant world. Very important, and the question will come of it definitely. A special branch of algae devoted to the study of diatoms is called diatomology. Okay? And diatomology, and these chloroplasts are discoid, plate-like stellate, contain chlorophyll A and C, same as that of ectocarpus we have. And the pigments is fucoxanthin, that is again that of same that type. But beside fucoxanthin, we have diatoxanthin because diatoms are present. And pyrenoids are naked. And the reserve food material is chrysolaminarin, which we did in the Xanthophyceae. See, I told you at that time, right? Chrysolaminarin. And then we have the one freshwater forms which we do in the practicals also. That is the navicula. See, navicula looks like this. Right? So we a lot of time identifies in the practicals navicula. Species, besides this, the species of Unia are epiphytic on the cladophora. So there is one species of cladophora and Oedogonium, these Unia species are epiphytic. And diatoms are unicellular and colonial. And this question will come, diatoms are unicellular, because I told you, one that is the, in case of Xanthophyceae, we have the multiflagellate and multinucleate. So now we are coming again back to the here. So cell wall is composed of silica. Mostly cell wall is composed of other items, but here it is silica. Very important. This question will come and called frustules. Frustules, frustules. The cell wall consists of basically the two overlapping theca. So it has two overlapping theca like this, right? Just like petri dish. Petri dish upper part we will take and lower part will remain there. That is, upper half is called epitheca and the lower half is hypotheca. Each theca is composed of two parts, valve and connecting band. And the question will come, a connecting band is called girdle. Now we are coming to the reproduction, vegetative reproduction. Here the vegetative reproduction occurs by means of a cell division. Very important. And the nucleus divides mitotically, and frustule, as I told you, just a silica part, increase its volume and two theca separates out. And two theca separate out and each can give rise to its daughter body. And then we have the asexual reproduction. And one more question comes here, statospores. Okay? And they are also known as hypnospores, endospores or cyst. And second is the oxospore. In navicula, the zygote forms a specialized spore called oxospore. It's very important. Also called restorative process. Because in this oxospores, the original size of the cell is restored. Right? So here it is very important to tell you that what will happen. The theoretical assumption of diminishing size of diatoms progenies was laid by MacDonald Fisher and called MacDonald-Fisher law, right? And the cell reach a minimum size range, typically 30-40% of its maximum size. So it is very important to you people to know that in diatoms, sexuality is a means of size restoration, right? That is why the oxospores, it is called restorative process, right? So then we are coming to the sexual reproduction. In Navicula, like we did earlier also, isogametes are formed and they fuse to form zygote. And this zygote contracts and it changes into the oxospore. So sexual reproduction is oogamous, and oxospores like same that of the asexual, but here these are the sexual. They increase in size, and then diploid nucleus in it breaks by mitotic division into the two, and one aborts and the two again divide mitotically to form two again. One again aborts and the second it will form a complete Navicula plant, right? This is the life cycle is basically predominantly by the diploid, that is called diplontic. The theoretical assumption I've already did, and I again tell you the sexuality is the means of size restoration. This is called restorative process. The last family of today we are doing is Euglenophyceae. Euglenophyceae is basically having a euglena unicellular, right, and possesses both animal and plant characters. It is not in much detail, whereas a slight bit questions will come. Here the Euglena sanguinea gives red coloration to the water due to presence of xanthophyll pigment called, this question will come, astaxanthe. The red color due to Euglena is due to the presence of a astaxanthin. The cell is naked without cell wall, and this is basically the plant character, but plasma lemma has outer covering. This is animal feature. The reserve food material is paramylon bodies, right? Now we are coming to the reproduction. So asexual, same as that of we did in Basilerophyceae, cell division. Nucleus, it divides mitotically into two, but here in that case one will abort and second will divide, but here the both will divide by longitudinal cell division, that is binary fission, and two individuals are formed. This is change it has. Sexual reproduction is more or less is absent, and here I just want to conclude with the other last type of the algae, that is called salmon. Salmon is basically algae occurring on the sandy beaches, which we did in third year also, Samosphere, and the examples are Vaucheria. We did just right, we finished it, and the other example is diatoms. So I'm again concluding what we did. So I just finished. So you have to revise again and again. So tomorrow I will come with the last and concluding part of the algae, that is consisting of a Rhodophyceae and Soenophyceae, right? So be with me, be indoor, be safe. Bye bye.